Jیووانی is a group leader and professor of physics at the University of Gothenburg. His research interest concerns both experimental and theoretical physics. He's conducting research on active matter, statistical mechanics of non-equilibrium systems, network theory, and deep learning. Today, he will talk on the interesting topic of the anomalous diffusion challenge. Jیووانی, thank you very much for accepting our invitation. You have 45 minutes for your talk. Please start your talk.
Okay, thank you very much. So, well, thank you very much for the invitation, and yeah, it's a pleasure to give this talk. Hopefully everything is working, just to confirm. So you see my screen and you hear me?
Yeah, everything is fine.
Perfect. Okay, so I will speak about this topic of the anomalous diffusion challenge, which we have organized recently. So I will give both an overview of the challenge itself, so what we did to organize the challenge, but also I will try to explain the physics behind anomalous, a bit of the physics about the anomalous diffusion and why it's interesting. So first of all, let's start with standard diffusion, and to do that we can look at a video, and this is just a video of a Brownian particle. This is a 4 micron diameter Brownian particle that is moving around. You see that it's just randomly moving, and it's a pure random motion. This is standard diffusion, the one that Einstein spoke about in 1905 in one of his famous papers. So why is there diffusion? The reason why there is diffusion is because a particle immersed in a fluid will be bombarded by the fluid molecules around it, like in this animation. So you will see that as the fluid molecules bombard the particles moves around. And if we don't know, even though this is completely deterministic, if we don't know, we cannot see the molecules, we don't really have any ways to predict the motion of this particle. And the resulting motion looks completely random, like in this trajectory. So this is the reason why we have a normal diffusion. And if you want to characterize normal diffusion, the standard way of doing that is by measuring the mean square displacement. So here we see a bunch of particles which are completely Brownian and diffuse. And as they diffuse, they move randomly. But what we can notice is that their average distance from the initial position grows linearly as this circle. So this means that we can characterize the mean square displacement. And if we do that, what we observe, now we measure also the mean square displacement, we observe that the mean square displacement grows linearly with time. So as time passes, the average distance, the average square distance that the particle travels from their origin will be linear in time. This is just a simulation with this number of particles. So this is essentially what characterizes standard normal diffusion. However, we can also think of cases in which the diffusion doesn't scale linearly. So we have the Brownian standard diffusion for scaling with alpha exponent alpha equal 1. So this means that it scales proportionally to time. But we can also have subdiffusive particles where the scaling is smaller than 1. So alpha is smaller than 1. So it scales like t to the alpha smaller than 1. In a limiting case, we can have a confined particle where the particle doesn't move at all, and then the scaling obviously would be alpha equals 0. And we can have super diffusive particles for which alpha is larger than 1, and in an extreme case, alpha will be equal to 2, which is essentially the case of a particle that is moving straight, basically a ballistic particle. So what produces these effects? What permits a particle to perform motion that is not standard diffusion? There are many reasons for that. For example, could be a crowding in the environment, so presence of other particles, the presence of traps in the environment, the heterogeneity in the environment, interactions, and so on. In general, any kind of deviation from a pure thermodynamic equilibrium can produce some kind of anomalous diffusion. And this is why it's so interesting in many different systems. So we see anomalous diffusion at all scales, going from atoms in some experiments with atoms in a optical lattice. So the diffusion of these atoms in this lattice will be subdiffusive. We can think of particles, molecules in the cytoplasm, which can be either trapped or actually, and therefore subdiffusive, or even transported and therefore superdiffusive. Of course, we can think of cell and bacteria, which move around, or also of animals, which in their foraging patterns might have superdiffusive motions. And it's interesting to notice that a few years ago, the Nobel Prize for Chemistry was awarded for work on studying and developing active molecules that can, in fact, present superdiffusive behaviors. And very importantly also, there was this Nobel Prize in chemistry, which was for the capability of actually looking at these molecules. So super resolution microscopy. So how do experiments usually work? Usually experiments work by measuring the position of the particles. So you acquire a video, for example, of molecules, you measure the position. Often this is done by using a centroid approach where the original image is thresholded, and then the centroid of each one of these white blobs is measured, and that is the position of the particle. And this then can be connected, linked into trajectories, which can then be analyzed. And this form of a quantitative microscopy to track particles has been around for a very long time. In particular, it goes down to at least 100 years. And here we go really back to the study of diffusion. In fact, Einstein predicted theoretically that diffusion would occur, and importantly, he also predicted that this would be a consequence of the existence of atoms, so of the granular nature of matter. And the experiment was actually done by Perrin in 1910, and consisted essentially in manually tracking microscopic particles, which were in this microscope, and measuring their diffusion. And by doing that, he basically proved the existence of atoms. So it's always a striking fact, at least for me, that just 100 years ago, people were not really convinced, scientists were still not convinced of the existence of atoms. They did believe that atoms were a good tool, mathematical tool to describe reality, but they were not convinced that they really existed yet. And Perrin, with this experiment, actually demonstrated it beyond any doubt. And by doing that, he actually got the Nobel Prize for it. Later, techniques to measure the position of molecules and particles improved in the 1950s, 60s, and so on. There was a lot of development in electronic means to measure the position of particles, and this developed into digital video microscopy in the 90s. And more recently, this led to the employment of deep learning tools to study microscopy images and to track particles. For example, we have also developed some software called DeepTrack. Now we are in version 2.1, in fact, which permits to do particle tracking and characterization, as well as other quantitative microscopy tasks. So you see that historically there has been a lot of interest in tracking particles, and this has had very important consequences for our understanding of reality. And anomalous diffusion has also been observed very soon when particle tracking started to be employed. In particular, there were techniques to make particle tracking already in the 80s to track single molecules, labeled molecules. And this led to the observation of some anomalous diffusion. And here you can see that these are early papers in which anomalous diffusion was observed, for example, in numerics, but also in biological interaction. One thing that is curious to notice is that here it's even written anomalous subdiffusion. So, subdiffusion is itself anomalous, but here, obviously, this result was so surprising for people at the time that to reinforce the idea that it was anomalous and subdiffusion. And this is not the only paper that shows that. Also, this other paper here has the term anomalous subdiffusion. So there was a lot of interest in subdiffusion. And the way in which these things, subdiffusion and anomalous diffusion behaviors have been characterized, has been essentially based on the study of the mean square displacement, as we have seen. So what does it mean, studying the mean square displacement? It means that you have to acquire the trajectories of many particles, calculate the mean square displacement of each of them, and then average these mean square displacements, and then calculate the exponent. So this is an ensemble average technique based on the mean square displacement. This is the standard in which way to characterize anomalous diffusion. However, you can see that it's not so simple, because you need to calculate a mean square displacement, and you need to have a lot of particles that you can ensemble average. Very recently, there have been several techniques that have been proposed just a few years ago. Several groups came up with the idea of using machine learning to characterize anomalous diffusion. And this led to several papers. So here you see some examples from very many different groups. One of this paper was also from my group in 2019. And the basic point here is that you can use machine learning techniques like neural networks and random forest to characterize the anomalous diffusion, even having a single trajectory, which means you don't need to do the ensemble average, you don't need to calculate explicitly the mean square displacement. So there were all these techniques coming up a few years ago. So the question at this point was, what is the best way to characterize the anomalous diffusion of a particle amongst all these possibilities? And that's where the idea of creating a challenge came up. So, as you might know, in science, especially in computer science, there are many challenges, which essentially are competitions, typically with some kind of prize, in which basically there is a task to be done, and whoever does it best wins the competition. There are plenty of this competition, like in platforms like Chegall or CodaLab and so on. So we thought that it was a good idea to try to make a competition to determine what methods are best to characterize anomalous diffusion. So, in fact, this is in a trend that has been very, very exploited in microscopy. So there have been plenty of competitions. Some examples of competition published in the last few years about different forms of microscopy and how it can be studied and interpreted. So this got, in particular, there is this competition which is based on the particle tracking challenge, where the purpose was to do the best particle tracking. Okay, so we decided then to organize such a scientific competition, and then to do that you need to do a series of steps. First of all, you need to have labeled data sets, then you need to decide how to host the competition, then you have to create a community that will compete, and finally you need to decide how to evaluate the competition, and finally you need to publish it if you want to have it in the scientific record. So you see that all these steps had to be addressed when we decided to make the competition. So we had to think about how to do all these things. So these are not easy steps, obviously. I mean, it's easier to say it than to actually do it. So let's start with the labeled data set. We have questions. So what data are we going to use? Real or simulated? How many dimensions? Under what conditions? Should we consider noise in the data? Which host to use? You know, there are many different hosts and they have different requirements. Should we use one of the standard platforms like Chegall or CodaLab, or should we have our own private web? How do we make the scoring, automatically or manually? Which community should we address? Should we go to physicists, biologists, computer scientists, AI people? And then how do we evaluate? You know, once you have the results from the various competitors, you have to decide who is the best. So the question is how to do that in a objective, as objective as possible way. And finally, obviously, you would like to publish it. And the question there is, how do we publish this? What happens if no journal accepts the final work? What do we tell to the people that competed, and so on? So a lot of questions to be addressed. So we started addressing these questions, and the first thing we decided to restrict ourselves to 5 models of anomalous diffusion. So there are plenty of mathematical models of anomalous diffusion, but the main ones are these ones. So there is the continuous time random walk, where essentially the idea here is that we have a random walk with obstacles, an annealed transient time random walk, which is basically a random walk where we have a switching of diffusivity over time, fractional Brownian motion, which is one of the most common models for viscoelastic system, which also show anomalous diffusion, scaled Brownian motion, and Levy walks, which are very much used in foraging theory and in the study of how animals look for food and for mates. So we decided to restrict ourselves to these 5 very important models. Then we decided to have 3 tasks. So one task was, given a trajectory, determine the value of alpha, so the value of the anomalous exponent. Second task, classify the model, tell which one of these models is the one for this specific motion. And finally, trajectory segmentation. So assuming that we have a trajectory that switches between 2 different kinds of diffusion, anomalous diffusion behaviors, we would like to identify when it changes and what are the anomalous diffusion exponents and the model before and after the switch. So how do we evaluate this? So in the inference of alpha, we use a mean absolute error. In the model classification, we decide to use an F1 score. And in the trajectory segmentation, we decided to use a root mean square error for the identification of the switching point, and then to make a ranking of the of the results that each competitor obtain on the identification of the switching point and on the identification of the alpha and model before and after the switch. So this is the 3 tasks and the relative metrics. Then we decided to implement this competition into 3 different, to generate simulated data sets for 3 dimensions. So 1D, 2D, and 3D, because they represent really different kind of anomalous diffusion that can be found experimentally. So for example, 1D, we can think of the motion of proteins along DNA, and in 2D, receptors in the plasma membrane, and 3D cells in a 3D matrix, for example. And all this work, all the coding and so on, was actually done by Gorka Munoz Gil, who was at the time a PhD student of Maciej Levenstein in ICFO in Barcelona, and now he moved to Austria. Okay, as a postdoc there. And okay, we also added some experimental data. So these experimental data were not used in the evaluation of in the ranking, and the reason is obvious, because we didn't really know the ground truth, so we cannot really say if someone is doing correctly or not. But the good thing is that all these experimental data were from published works in which the anomalous diffusion had been studied independently from us. So we could use this experimental data to validate the various algorithms. So the question was, will our algorithms that are not optimized for this experimental data in any way give us something that is comparable to what people have found in the literature? And will the algorithms from the different competitors agree with each other? So all these questions could be addressed by having some experimental data too. Sometimes this is called the reality gap. Okay, so let's see the timeline of the competition. So the idea was actually born Carlo Manzo came up with the idea from the University of Vic in February 2019. And then he recruited the team of Maciej Levenstein. And then he recruited also myself and Ralph Metzler. And from then we started having weekly meetings to discuss how to organize things that lasted for like 6 months. And until we managed to open the competition in March 2020, went through our first phase, which was pretty long. It lasted until September 2020, in which we basically recruited teams. They could compete, but they could compete and see their scores, but it was just the first phase where we wanted to attract as many teams as possible. Then we went to phase 2 until October 2020, in which the team were competing, and the ones that managed to submit good results in phase 2 went to phase 3 and competed for 1 week for the real final ranking, which closed on November 1st, 2020. After that, we had the winner announcement 1 week later. We had a wrap-up paper written as you can see, it took 1 year to write up the paper. And we organized a workshop, which was done last December in Barcelona. And finally, a special issue for where we collect all the contribution from the participating teams. Okay, so this gives you a timeline of the of the competition. So how many teams participated? There were like 16 teams for 14 institutions that participated and competed. And here you see the list. I will not go through the list. And then we went, I will show you a bit of the results. So task 1, here we calculate, we have the mean absolute error for the inference task. And you see, of course, it's better to have a low value because it means that you have a lower mean absolute error. And we focus on the 1D case. And what we see is that all the top teams basically employed machine learning techniques, while standard statistical analysis was really low in this ranking, which is not the fault of the teams. Probably means that the machine learning can really see something more than standard mean square displacement analysis in these trajectories. And here we can see, for example, the ground truth exponent against the predicted exponent for the best method, and you see that it does pretty well. And compare with what you obtain with the mean square displacement. So you see that the results of the machine learning are much better, in fact. And here you see how the various teams performed as a function of the underlying model, and you see that ATTM and SPM are a bit more difficult to analyze. Okay. And here you see that as a function of length of the trajectory, of course, you get a better result, which is not unexpected. But the good thing here is that the time average mean square displacement saturates pretty fast after a trajectory of 200 points, while machine learning can really improve for much longer. So it keeps on improving until at least 500-600 points. Okay. And here you can then see that also, as you can also expect, as a function of the signal-to-noise ratio, we obviously get better results when the signal-to-noise ratio is higher, so we have more signal compared to noise. Okay, the second task was classification task. We use a F1 score, and here, of course, you get better result if the score is higher. Again, we focus on the 1D case, and we see again that the mean machine learning method perform better than the statistical analysis methods. And here you see the ground truth versus the F1 score as a function of the alpha of the ground truth. You see that, you see some interesting facts. One is that for alpha close to 0, which practically means we are going towards particles that are more immotile, things get a bit worse, which is to be expected. The particle move less, there is less to see. And also for alpha equal 1, you get a bit worse results than in other cases. And the reason also here can be understood easily, because for alpha equal 1, we have a diffusive motion. And a lot of those models that I indicated before converge to diffusion, normal diffusion for alpha equal 1. So it's actually difficult or almost impossible to distinguish between them. So it makes sense that there are drops at 0 at 1. Again, things improve as we have longer trajectories, not unexpected. And again, things get better as we have less noise, which again is not unexpected. And here we can see a nice plot of the ground truth versus the predicted model. This is a typical plot that you will see basically everywhere. If you do any kind of machine learning, this is a confusion matrix, where basically we show how a ground truth is classified, how different ground truth models are classified into different predicted models. This helps you to see which one is the most difficult to classify. Here you see that, for example, TTM is one of the most difficult to classify, while continuous time random walk seems to be classified pretty easily, as well as Levy walks. Okay. And finally, we have the third task, which is the segmentation task. So in this task, what we want to do is to find the switching point as well as the values of the alpha and the model before and after the switching. We didn't have many teams participating in this task. There were only 3 teams because probably it was the most difficult task. So the results are obviously less, we have less confidence, but again, we can say the machine learning works best. And we can say that we can see that the alpha for the first and second segment, it's classified. We cannot really say much more than that in this case, because there were not enough teams to actually conclude something completely definitive. But what we can conclude between these 3 teams is that the inference results were similar between them, and they mainly had, some of the teams had problems in classifying the different motions. If you want to play with this, or if you have your own algorithms and you want to test them against the competitors of the challenge, we have an online interactive tool in AnDiChallenge.org/interactive tool, where you can explore the data further. You can look at data, for example, I want to see what is the performance of the different methods for a certain level of signal to noise ratio and certain length of trajectory. You can do that and see which one is the best for that case. But you can also upload your own results and see how they would score against all the competitors of the challenge. And here you can see the screen for the inference task, for the classification task, and for the segmentation task. So you can, for example, check what happens for short trajectories and see that for short trajectories, the Bayesian inference stands here, and so on. You can do a lot of things. Okay, finally, we tested this method on experimental datasets, and these are different experimental datasets. As I said, they had been already analyzed in the literature, so we didn't collect the dataset because we wanted to have someone else to analyze them independently from us. So in this case, we have MRI molecules diffusing in bacteria, inside the bacteria. And the original results that were gotten by the original paper was that these molecules are subdiffusive with alpha between 0.7 and 0.77, and they are between continuous time random walk and fractional Brownian motion. So what we did was to take these trajectories and pass them through the algorithms, that the top 5 algorithms. And these are the results we got. And what you see here is that these 5 algorithms perform pretty well, obtaining more or less the value of alpha that would be expected, which is this dashed line. And they perform better than just applying the time average mean square displacement, which will give this gray result. And also, we observed that one thing that is interesting to observe is that all the algorithms classify these motions confidently as ATTM, which is different from the continuous time random walk in the original paper. But to be fair, it was not considered in the original paper, so we don't really know whether the original paper authors would have concluded this was an ATTM if they had considered such a motion as a possibility in their original analysis. What is nice is that, in agreement with the original paper, also these algorithms conclude that this is not a fractional Brownian motion. As another example, we can think of the motion of telomeres inside the nucleus of the cell. And here, the original results was that there was alpha equal 0.5 short times that went towards alpha equal 1 for longer times. And this was a fractional Brownian motion according to the original work. So what we get, which is nice, is that we get, in fact, the results fit within the two 0, 5, and 1, as in agreement with the original work. And in this case, we also obtain that also our models predict that this should be fractional Brownian motion in agreement with the original work, which again is a nice check that the algorithms in fact work also for real data. And as a next example, we have receptors moving in a membrane, in a plasma membrane. Here, the original value was found to be 0.84, and this was determined to be a TTM. And the results again agree very well with the original work. And in fact, also the classification returns an ATTM, which is again a nice result. And finally, we have the motion of atoms in a lattice. This is very interesting because, well, first of all, we are in a completely length scale, different length scale. And secondly, these atoms make very short trajectories, like 10, 20 data points. So it's a very challenging regime. And we will see that, that in the results. So first of all, in the original work, this was predicted to be alpha equal 1, so pure diffusion with continuous time around the walk dynamics. And what you obtain is that it's compatible with the pure diffusion. And we obtain also a continuous time around the walk dynamic with high probability. We have to say that in this case, again, the trajectories are very short, so it's very difficult to do the classification and the inference. And also, since alpha is equal 1, it's very difficult to classify the model, as I said before, because all models tend to converge to something very similar when alpha is equal 1, so for the case of standard diffusion. Okay, so we can then draw some general conclusion from this challenge. First of all, is that basically, almost all the methods that were proposed by the participant outperform the standard time average mean square displacement approach, which is what anyone would use as a first attempt. So basically, almost anything will do better than that. And machine, and in particular deep learning method, improve enormously the precision of inference of alpha. Okay, especially, and this is especially true for short and noisy trajectories, which is not totally unexpected. In model classification, we have a similar results. So, well, one of the results is that the similar result, machine learning and deep learning works much better than standard methods. So it's worth considering, especially for short and noisy trajectories. And one thing that is that was brought up during the challenge, and it's a good point, is that we choose 5 models. Of course, the nature might not uniform itself to these 5 models, could be something different, which is fair. So it would be nice to develop methods in which there is also a none of the above class, where certain trajectory can also be classified as none of the above, so not classified essentially. And finally, in the segmentation task, this, as I said before, we had very few participants, so we cannot really claim to have final conclusions. But yeah, it's quite challenging as a task. That's why probably few participants took it on. And therefore, it's nice to explore this further in the future. And what we observed also is that there was some asymmetry in the determination of the diffusion constants, exponents, and models, underlying models for the first and second segment of before and after the switching, which might make sense, but we need to understand better why so. And finally, not surprisingly, but pleasingly, we observed that the methods that were developed on the simulated data to compete on simulated data work well also to classify and to analyze experimental data on which they were absolutely not trained, obtaining results largely in agreement with what was in the literature. And very nicely in agreement between the different methods. So all methods returned results that were very similar to each other, which again gives a lot of confidence in applying this method to experimental data. Okay, so this work is published, was published, as I said, last year. So it was published in Nature Communication. And all people that took part in the challenge until the end were co-authors in this. The organizers were the idea originated from Carlo Manzo, as I said, at the University of Vic, and he worked very closely with Gorka Muñoz-Gil, who was at the NIC for Barcelona, but now he's in Austria as a postdoc, and he's the one who really did the hard work of coding everything. And then there was Miguel Ángel Garcia Marc at the Polytechnic University of Valencia, Maciej Levenstein, Ralph Metzler at Potsdam, and myself at Gothenburg. And we were the 6 organizers, while all the others were the participants. So what's next? Well, then we had also this very nice special issue, which is now essentially closed, and to which there were many different lots of the participants in the challenge actually accepted to submit a paper to this special issue explaining their technique in detail so that people can reproduce it. So if you're interested in the topic, this is a very good resource. And as I said, it's almost closed now. And what's next? Well, what's next is that we are now organizing the second challenge. So we will go with AnDi 2, the Anomalous Diffusion Challenge 2, in which we will focus much more on biologically relevant problems in 2 ways. First of all, we will work much more on molecules or particles that change behavior. So let's say, in a sense, our focus will be more on expanding on task 3. And the second focus on biology is because we will also study not only trajectories, but also videos, simulated videos of particles to see how much information can be extracted from the original data that actually people usually have, which are, as I said before, videos. So if you're interested, obviously, you are all welcome to participate in this challenge. Like last time, it will have different phases, and we will have a workshop, special issue, and a final publication with all participants. All participants that obviously arrive at least high enough in the challenge, not just submit once. Okay, so, but please think about it. If any of you is interested in this kind of topic, it's a very good way to connect with an international community interested in the topic and also get some publication out of it that helps anyone's curriculum. Okay, with that, I would like to conclude. So this is a picture of my group and not only my group, my group and 2 other groups a few weeks ago after a long time in which we could not take pictures because of COVID, and these are the funding. And with this, I would like to just wrap up. So what I try to show you is that anomalous diffusion is very interesting and has a lot of potential to understand nature and how things occur at the macro scale, and also that there is a lot of potential in employing competitions to basically to guide the scientific community towards a certain topic and to be able to explore this topic more in depth. And with that, I would like to thank you, and if you have any questions, I'm happy to take them.
Thank you, Giovanni, for this super interesting talk. We have time for some questions. Please raise your hand. Dr. Kolachi, please ask your question.
How about now? My question is about a possible anomalous diffusion that requires a very long time to detect. Could that be one of these 5 types, or maybe a mixture of them, or how can you make sure that you have waited long enough to find the alpha, for example?
That's a good question, in fact, because one thing that In anomalous diffusion, in general, anomalous diffusion means for a certain time scale in reality, because most physical system will, in the very long, I mean, if you go to very, very long time scales, most physical system will be either diffusive or confined. Real system, I mean. So when we speak of anomalous diffusion, often we really imply a certain time scale. Then how do you know that you are waiting long enough? I don't know. Actually, I don't have a good answer for that. I think the main point is that you can really claim that you have anomalous diffusion for a certain time scale more than in absolute terms. It's the same thing with the Levy flights. The real system will never have a real Levy flight simply because it's impossible, or white noise for that matter.
Okay. Thank you very much. Thanks.
If there is any other question, please raise your hands. Giovanni, can I ask my own question? Having
Sure.
correlation between, different directions can make the diffusion anomalous. Do you agree or not?
Can you repeat? Sorry.
Having correlation between different directions. For example, if a diffusing particle near a flexible membrane, can it make the diffusion anomalous or not?
Yes, the presence of boundaries and structures in the environment or heterogeneity in the environment is one of the causes that can produce anomalous diffusion from a diffusive system.
Mm-hmm. Do you have any program?
Sorry, can you repeat?
Do you have any program to consider such kind of,
Yeah. Actually, in the second challenge, which we are organizing right now, we also consider heterogeneities in the environment that then produce some kind of anomalous diffusion.
Mm-hmm.
But but that's definitely, also there are obviously in the challenge we will consider something because we have to choose what to do in the challenge. It cannot be everything, but in general, the presence of heterogeneity in the environment can produce anomalous diffusion.
Thank you. It seems that there is no question. Finally, I would like to thank you again and say goodbye to you.
Thank you. Bye.